Thank you for attending today's webinar, Genomic Tools to Validate New Generation Conditionally Reprogrammed Tumor Cell Line Models, presented by Dr. Seema Agarwal. Dr. Agarwal received a Ph.D. in Organic Chemistry from the University of Allahabad, India, and a second Ph.D. in Biochemistry on chromatin structure from Lehigh University. Her postdoctoral work was done in the labs of Dr. Michael Snyder and Dr. Shirlene Roeder at Yale University, where she worked in classical genetics and genomics. Dr. Agarwal had a brief stay in a biotech company where she led a group on cancer drug target discovery. She joined Yale University as a research faculty in 2004 to work on different aspects of cancer biology, including tumor-specific protein and microRNA as biomarkers. Dr. Agarwal moved to Georgetown University in 2014 as an associate professor. Her lab is currently focused on developing conditionally reprogrammed in vitro and in vivo zebrafish metastasis models for several tumor types, including rare tumors, to study tumorigenesis and metastasis. Without further ado, here is Dr. Seema Agarwal.
Thanks, Eric, for giving me an opportunity to present my work and for a nice introduction. And thanks also to GeneWays to approach me for this presentation. As Eric said, that I will be talking about how to integrate the genomic tools with next generation conditionally reprogrammed tumor cell line model system. The outline of my today's talk will be basic in vitro cell culture models. I will talk about the advantages and drawbacks of the conventional cell lines model system. I will introduce the conditional reprogramming cell technologies to establish next generation patient-derived cell cultures that I will refer often as CR. I will give examples throughout my talk to discuss the need for proper authentication and validation of these patient-derived cultures using various genomic tools, and some of them are mentioned here, and how when the genomic tools is combined with CR technology is used to rapidly identify the drug targets as well as the drug therapy that has already been in clinical use. From a long time, scientists have been trying to establish bona fide models for growing and studying primary tumor outside of the human body. The first very human tumor cell lines, as we all know, was established as HeLa cells at Johns Hopkins University in 1956. This very first tumor cell line was from a high-grade cervical cancer tissue material from Henrietta lacks. HeLa is a HPV positive tumor cells, which is allowed or rather paved the way for its establishment. Since then, only a couple of thousand tumor cultures have been established because it has been still very difficult to rapidly and robustly establish the tumor cultures directly from patient samples. And not only that, unfortunately, most of these conventional tumor cell lines are from high grade and often from patients which have been diagnosed with metastasis. And all tumor cells are spontaneously immortalized. However, to establish normal cell cultures in dish has been even more challenging, and only a handful of normal cell lines exist to date. They all have been immortalized using exogenous reagents, such as overexpression of hTERT. An example is HME1 hTERT, which is a memory epithelial cells. Using the SV40T large antigen, HMEC, which is also a memory epithelial cell lines, was established that way. Oftentimes, people have used the viral HPV, human papilloma virus, E6, E7 proteins, and it has been used extensively to establish primary keratinocytes. And another example is use of c-MYC to immortalize the normal cells. One can immortalize the normal cells and establish cultures using any of these methods or other methods that are not mentioned here, but they often lead to the neoplastic transformation of these immortalized normal cells. So they have a limited use of what it can be used for basic and translational research. As I said earlier, that only a handful of tumor cell lines exist, and most of them are clonal in nature. And what do I mean by clonal? Which means that tumor itself is a genomically very heterogeneous, but these tumors, conventional tumor cell lines, are only clonal in nature, meaning that all cells have the same genetic alterations. So they are not heterogeneous, but rather homogeneous cultures, which is a really big problem as we are moving towards the personalized medicine era, where we really need a method to robust and rapidly establish a patient-derived cell models, which can be rapidly combined with the genomic information from patient to dictate the personalized therapy opportunities. So to overcome these limitations, Dr. Richard Schlegel from Georgetown University have pioneered a technology which he published in 2012, in which he uses the Rho kinase inhibitor and combined it with the mouse fibroblast cells as a feeder cells to induce the conditional reprogramming of tumor and normal epithelial cells. In that seminal paper, they have shown that both feeders and Rho kinase inhibitor are needed for indefinite culturing of these cells or expansion of normal and tumor cells. If the Rho kinase inhibitor is out and you have only feeder cells, the cells will grow for a certain passages and then they stop growing. If the feeder cells are out and then only the Rho kinase inhibitor is present, then also the same phenomena will happen, that the cells senesce and they stop growing. And in the media which exist for the prostate epithelial growth media or memory epithelial growth media that one can buy from different vendors, they allow the cells to grow only like 5 or 10 passages after that cell senescence stop. In that paper, they also showed that these cells were stable cell lines. They did not create any genetic alterations. They kept the karyotyping. They showed that these cells at the different passages, passage 2 or passage 33, have maintained their DNA fingerprinting, which means they are not contaminated with any cell lines, as has been often found. It is a very increased efficiency. These cultures can be established, and no additional immortalization is required. So only the irradiated feeder cells and the Rho kinase inhibitor in a defined media allow these cells to grow indefinitely. So how does it work? So this shows us schematically that you can use a surgical specimen, core biopsies, fine needle aspirates, blood, urine, PDX tissue, which is patient-derived xenograft tissue material. And oftentimes for surgical specimens, core biopsies, it is required to have a pathology evaluation just to ensure that in the tissue material that has been used to create the cell culture contain a lot of tumor cells, at least 90% of tumor cells. Tissue is diced, enzymatically digested, and then J2 fibroblast, which is embryonic mouse fibroblast cells, have been trypsinized and irradiated at 30 gray. And then when this is combined with the digested tissue in the presence of Rho kinase inhibitors, cells grow indefinitely in this condition. And a detailed method of how to grow different tissue types is shown we just recently published in Nature Protocol. Because even though this overall idea worked, but for different tissue types, we needed to tweak the conditions to make sure that the cells will grow. So the details of how to grow a different tissue types, example, breast, lung, prostate, salivary, check the Nature Protocol paper and you will get the information that you will need. And this just shows that how rapidly the cell cultures are established. This is a 3-day movie in which these are epithelial cells and these are the irradiated mouse fibroblast. Yes. As the cells grow, the epithelial cells will push out the fibroblast away from the colonies, and the colony which is adjacent to it, they will merge together as this fibroblast is pushed out of the colony, and they become a very large colony within 3-4 days. So it is very rapid, it is very efficient, and it is very robust, and that is what we needed for a patient-derived tissue cultures. So now in my rest of my talk, I will give you several examples of how this technology can be used for rare tumors and how it is really important to combine the genomic tools. This is an example which is published from Dr. Schlegel's group again, which is for a very rare kind of tumor in which a patient who was 23-year-old male was diagnosed with recurrent respiratory papillomatosis. In short, it is called the RRP. This poor fellow has gone through 350 surgeries to control disease. And finally, this was extended into the trachea and bronchi and ended up making 3 sentinel tumors in the lung, which were increasing by 21%, 72%, and 130% in the preceding 11 months. Georgetown lab received both the lung tumor specimen as well as the adjacent normal tissue, and they were able to create the cultures using the CR technology both for tumor and normal. And that was done within the 3 days. They had enough cells to figure it out what would be the target. These RRPs are infected with HPV. They are HPV driven. So they used the genomic tool, in this case the PCR technique, by using a different primer sets for HPV to identify that HPV11 was present in the sample, and which is done within the day 5, and in day 8 they did the in vitro testing of chemosensitivity assay, identified a drug, and the patient was treated with that drug, and is shown here in 2011 that patient was treated with the drug identified using the CR technology, and in April 28 the tumor has been shrank and it was stabilized for 2-3 years until the patient passed away. So this showed that how quickly, within less than 1 year of establishing this technology, that it has been used in the clinical setting. Second example of rare tumor is neuroendocrine cervical cancer. This is the only cell lines that exist to date. This was again from a female patient who was diagnosed with neuroendocrine cervical cancer. The tissue was obtained at the Georgetown lab. The CR culture was established, as well as the 3D spheroid cultures. This was a very aggressive tumor. It created a xenograft tumor in mice very rapidly within a month. And these cultures in vitro shows the typical neuroendocrine marker of the presence of high expression of MYC and somatostatin, as well as some other markers. So not only the proteomics was used to validate these cells, but also the genomic tools. So neuroendocrine tumor was tested for the HPV, and it was found to be HPV16. And then they have used the quantitative PCR technology to identify the copy number of HPV16, and they found that it is about, on average, 50 copy number in the cells growing in the dish. But when they used these cells in mice to create the xenograft tumors, they found that the copy number of HPV16 has increased to about 100 HPV16 copy numbers. Then another genomic tool which has been very useful in this particular tumor was the FISH. fluorescence in situ hybridization. So the question was to where the HPV16 is integrated into the genome, and when they used the MYC and HPV16 specific markers, as is shown here, that HPV16 is integrated into the MYC locus. And oftentimes, or most of the time, when the tumor is infected with HPV, then it is driven solely by the HPV. But in this case, it was found that this tumor was no longer dependent on HPV16 for survival, but rather dependent on the MYC gene. So this technology, along with the genomic tools, identified the MYC as a target for this specific tumor. Now, moving on to the rare tumors, my lab has created or established first authenticated cell line for adenoid cystic carcinoma of salivary gland. This is such a rare tumor that we could not get the fresh tissue, but University of Virginia had created about a dozen patient-derived xenografts. So we received the PDX tissue material from 10 individual patients, and we established 9 out of the 10 individual CR cultures. Just to give it a little bit information about why it is important to create the adenoid cystic carcinoma of salivary gland when it is so rare, because it is a very rare and very aggressive tumor. The 5-year survival rate is only 45%. There is no targeted therapy exist. This tumor is treated only by the radiation currently. And this tumor is known to have a MYB gene translocated in 60 to 80% of tumors, and in others, the MYB L1 translocation has been identified. So this tumor model will be very useful to know whether MYB is the driver for the tumorigenesis in or for metastasis or not. And for this reason, we created these cell lines. So as I said, we created 9 out of the 10 individual CR cultures, but unfortunately only 2 out of the 9 were able to survive beyond 5 passages under the CR conditions, and other differentiated after 3-4 passages. And it's not that if you look into the literature, you will find that there are a handful of ACC cell lines exist, but this paper brings out the issue of how the genomic tools should be used and why the genomic tools should be used to validate and authenticate the cell lines. Because this shows that all the cell lines that exist in the literature are either contaminated with HeLa cells, which have been found after doing the DNA fingerprinting of these cells, or they were not even human cells. 2 of them were either mouse or rat cells, and then the other few cell lines were misclassified because they were either urinary bladder cancer cells, or they were have some other contamination like HeLa cells. So again, it brings out the issue that how it is really important to use the DNA fingerprinting and other genomic tools to validate the patient-derived cultures. So since we have created 9 out of 10 cultures, we tested all of them by DNA fingerprinting using the PDX tissue as the gold standard in each case. And as it's shown in this, that are all 9 cultures, even though some of them have a very low passages, they all match perfectly with the tissue of origin. So one of the cell culture, ACC11, we went further and validated this culture because, as I mentioned earlier, that MYB is translocated in these. So we checked the MYB split probe. MYB is located in chromosome 6, and as is shown here, that there is these 2 are overlapping here, which means that this is an intact chromosome, and MYB is in the chromosome 6 and not translocated. But in this chromosome, MIB is translocated because these green and red spots are far apart from each other. MIB is often translocated to chromosome 9 to NFIB gene, and so we use the MIB NFIB primers to check whether it is expressed at the RNA level, and it is shown here, the real-time PCR followed by the regular PCR using the primers for MIB or NFIB. We checked by the protein, and we showed that the MIB NFIB fusion protein, which is shorter in length, is expressed. Furthermore, we did the spectral karyotyping. It is a very useful genomic tool to look at the translocations and the chromosomal copy numbers. This shows that ACC11 is diploid in nature, but it has a 6-9 translocation, which means MIB gene is translocated to chromosome 9-6. But it also has 2 other mutations, chromosome 2 to 11 and 11-22 and 2 to 22, which is a balanced translocations which have not been known before for this tumor. Another ACC tumor has a different translocation. It has a 6-1 translocation in which MIB is translocated to TGF-beta R3 locus, which was further confirmed by fluorescent in situ hybridization using the probe specific to MIB and TGF-beta R3. Then we contracted it out to GeneWiz to use their 48 TrueSeq cancer panel to sequence the ACC11 tumor, and we found that it has the mutation in ATM gene as well as in FGFR2 gene. And these mutations in the cell lines were also validated in the PDX tissue, because it could be possible that under the growth conditions in the dish that the cell cultures will acquire any mutation. So it was really important for us to make sure that these mutations that we identified using the GeneWiz 48 TrueSeq cancer panel is also present in the PDX tissue. And as shown here, that is the case. And in both cases, in the PDX tissue as well as in cell line, it's a heterozygous mutation in both the cases. So again, genomic tools was very useful to identify the novel mutations, novel translocations for salivary tumors. And this led to the identification of regorafenib, which is a multi-kinase inhibitor, as well as the VEGF inhibitor, as a potential therapeutic target for ACC, which was validated rapidly not only in the cell lines, but also in xerograft and also in using the zebrafish model system, which is not the focus of this talk, so I will not go in detail, but we have already published a paper in which, if you need the detail, you can go and check it out. So this shows that how CR technology is useful in creating cultures for rare tumors for which not much information exists, and how the genomic tools can be used to rapidly authenticate and validate these rare tumor cultures, and then to use to identify the drug targets, and which will lead to the drug therapy. Now, earlier I have mentioned that all the conventional cell lines that exist, they are clonal in nature and they lose the heterogeneity of the tumor. So it was really important for us that before this conditional reprogram, tumor cultures can be used for drug target identification or potential drug therapy to see whether the heterogeneity of the tumor is maintained in these cultures or not. To do this, we recently published a paper on that. You can get all the details in it. We created 10 non-small cell lung cancer conditionally reprogrammed cell lines. And to our surprise, we saw that the morphology of these tumors are maintained. As an example, this is one patient which was squamous cell carcinoma. Squamous cell carcinoma has the morphology of very large cells with very small nuclei and large cytoplasm, and that is shown here, and they are arranged in a puzzle formation, which is the H&E of the tissue itself, and you can see the size of the cells are really big. In contrast, adenoid cystic carcinoma of non-small cell lung cancers are really cobblestone-like cells with a very large nuclei and very small cytoplasm. And this H&E, you can see that that is the case, and in the CK and in the H&E tissue too. And CK7 is cytokeratin 7 is a marker for lung cancer, and it has been shown here to be present in these CR cultures. And these squamous cell carcinomas have a very prominent desmosome, so I'm not so sure whether you can see it or not, but desmosome structures were also maintained, and you can see it more clearly here in the squamous cell carcinoma CR cultures. So to test whether they are heterogeneous culture or not, the first thing we did that we look at the copy number variations. Because lung tumors have a lot of copy number variations, we used the frozen tumor tissue material, and we have also created the adjacent normal CR cell line. So we used that, and we used the tumor CRC line. And as shown here that frozen tumor tissue samples and the tumor cell lines match quite well from each other, while normal CRC line showed very few copy number alterations as expected, because normal cells should not have many copy number variations. Then next, we did the whole exome sequencing of 10 individual samples and in triplets, meaning they had the primary tumor tissue, normal lymph node tissue from the patient, and tumor CR cells. The first thing we did is to do the Jaggard plot to identify the percentage of normal and tumor in the culture. So when you take the tumor from the patient, tumor cells are growing within the normal cells. So it is inevitable that you will get both normal and tumor cells. And this, since CR technology allowed the growth of both normal and tumor cells, it was necessary for us to check that in each tumor cell lines, how much normal and tumor is there. And as the Jaggard plot shows, that most of the cell lines are grouped together and clustered together in this region, which shows that about 50% of normal cells and about 50% of tumor cells are present in these cultures, except in this one, in which the tumor cell population is very, very low. It is mostly normal. So if, as an example, if they were all normal, they will be in this corner, in the right side corner, bottom corner of the graph. And if they are all 100% tumor, they should be at the top left corner of the graph. The next thing we checked, that based on the number of single nucleotide variations, SNVs, that what is the relationship with the normal tissue and with the tumor tissue. And here it shows that about 94.7% it's matching with the normal, and only 5.22% the CR culture is different from the normal. While in the tumor, is more than 98% is similar to the tumor SNVs, and only 1.57% is different, which means that the tumor cells or tumor cultures look more like a tumor here than normal. Then using the SNVs, we derive the mutant allele tumor heterogeneity score, and this is the main factor of finding out how heterogeneous the culture is. And here it shows that the CR tumor cultures are maintaining the heterogeneity of the tumor more than 90%, and in some cases it is 100%. And then, as an example, is this samples where the tumor and the CR culture, which is mentioned here as the CR tumor, are overlapping with each other. And again, this one is also more than like 98% overlap. But in some, there is a vast difference. But nonetheless, all of them are heterogeneous in culture. None of them are clonal, even though there are some differences between the tumor and the CR cultures. And those differences could be for different reasons. It could be because the tumor is so heterogeneous that the piece of tissue that we use to sequence is different from the piece of tissue that we use to culture the cells. It could be that CR cultures preferably grew some clonal populations but leave out some other clonal populations, and they did not grow. But nonetheless, it shows that CR cultures are very heterogeneous in nature, so they can be a really good model system for basic and translational research, for identification of the drug targets, and moving towards the personalized medicine approach. Furthermore, we checked whether the heterogeneity of the primary tumor maintained in the cell lines by doing the principal component analysis. And here in this principal component analysis, you can see that all triplet, the CR cultures, the normal, which is green, and the tumor, that is blue, is clustered together, which means that these CR cultures maintain the genetic features of the patient. Because if they were clustered with some other patient, then it would be not mimicking the genetic features or genetic makeup of the patient. On the right in the B, we are looking only into the non-synonymous SNVs, and the red or orange circle is the conditional reprogrammed cells, and the blue is the primary tumor tissue from the patient. And as you can see that most of them have a very high overlap. It is more than 90% overlap of SNVs. Only there are some few differences in each cases. And these differences again can be for several reasons. One can be the piece of tissue that is sequenced, as I mentioned earlier, and the piece of tissue that is used to establish the CR cultures are different. And secondly, it could be possible that some of the clonal population in the tumor, which were present in a very, very low percentage, so in whole exome sequencing, those clonal population went into the background, but they are able to grow up in the CR cultures. So in this way, we can really identify what are these mutations here in which genes, and can they be the reason for metastasis. And that is not a far-fetched thing, because recently, as we published our paper, a paper came out from a different group from Illinois, which showed that in the case of the breast cancer patients, they have used the CR technology to establish the cultures from primary breast cancer tissue as well as from the metastatic tumors. And when they did the whole exome sequencing, and they found that CR culture was able to expand the minor clonal population both from the primary tumor and metastatic tumors. And this way they could find the actionable drug targets. And this is the table from the same paper, which shows that the cultured tumor cells were able to identify the mutation in genes which can be targetable, which is shown in the bold colors, bold letters here, both in the primary tumor as well as in the metastatic tumors. And these are the mutations found in the culture cells which were not present in the tumor. So this way they were able to identify the mutations in the genes which can be a potential drug targets, and they have done more analysis on these mutations, and to show that yes, indeed, targeting these mutations can either stop the tumor formation in mouse, or it can stop the metastasis of the tumor. Another example that I'm showing about the heterogeneity of the tumor is patient-derived pancreatic cancer conditionally reprogrammed cells. And this is an unpublished work from my lab, and we have established 8 different pancreatic cell cultures, but I'm showing you only one of the example of it, where we have employed the spectral karyotyping genomic tool to look at the tumor heterogeneity of it. And as it can be shown in these 2 different sky metaphases nuclei from the same patient or CR cell cultures, they are very different from each other, not only for translocation, but also for the chromosome number. As an example, this the bottom sky metaphase has 3 copy of chromosome 1, while in the top left one has only 1 copy of it. It lost the 1 copy of the chromosome 1. And then the translocations are very different here too. Here is a 2n18 translocation, which has been found 2n18 in the second metaphase spread 2, but it has also found a different translocation also present, which is involving only chromosome 1 or chromosome 4 in this case. And like that, there throughout you will see that there are differences in the chromosome numbers as well as in the type of translocations that exist, which shows that the CR cultures are highly heterogeneous, which cannot be appreciated without having the cultures, because the primary tumors or tissue cannot undergo the spectral karyotyping. Spectral karyotyping can only be done in the cells which are growing in the dishes. And so by doing the CR cultures, we were able to see that how vast the differences are in the patient-derived pancreatic cancer cells, and that we are using these to look at the sensitivity of the these pancreatic cultures to known drug gemcitabine that is used clinically to treat pancreatic tumors. And we are using it further to identify some other drug targets. So before this is my sort of a final scientific slide, and it shows that my vision of how the patient-derived toolkit can be established very rapidly and reliably to move towards a personalized medicine approach. And this slide shows that you can start with fresh tissue or from mouse patient-derived xenografts and create the cultures using the conditionally reprogrammed technology, and how before they can be used for any applications, some of them are mentioned here, they have to be authenticated and validated using the genomic tools. And there are a few genomic tools that are mentioned here: DNA fingerprinting, karyotyping, and FISH. And obviously the next generation sequencing also can be employed, as well as many other genomic tools. So I hope I was able to impress upon you that how the conditional reprogramming technology go hand in hand with the genomic tools that are available to us to move this system to a personalized medicine era. With that, I will end my talk and show you the scientific collaborations for the center at the Georgetown University on conditional reprogramming. The director is Dr. Richard Schlegel, and we have a various collaborations ongoing with different institutions. This is my lab, which shows the past and present members of the lab. Alex Lekan is not in the main picture because he's a recent addition to my lab. And with that, I will end my talk, and thank you for attention, and I will be happy to move towards the Q&A session.
Thank you, Seema. That was extremely interesting. We're now going to move to the Q&A portion of this webinar. If you have a question, simply type it in under the Q&A tab. A few came in during your presentation. The first one is, During the talk, you mentioned that lung tumor CR cultures are contaminated with normal cells. Is this a general phenomenon in CR technology? If so, would it pose a problem in moving this technology to clinical settings?
Yeah, it's an excellent question. Yes and no, the short answer is. Definitely, as I said earlier, that CR technology has been used or allow both normal and tumor cells to grow in dish. So anytime a tumor is tissue is procured for cell culture, both if there is a lot of normal cells, they will grow in the dish. So yes, the tumor cells will always most likely will be contaminated with the normal cells. So that's why I also mentioned earlier that it is really important to do a pathology evaluation of it before it has been used to establish the cultures. So the phrase is that what you garbage in and garbage out. That if you procure the tissue sample, a tumor tissue sample, which has 90% of normal and only 10% of tumor, one cannot expect to grow the tumor out of it. You will grow mostly the normal cells from it. So it is really important to use the pathology to make sure that you have at least 90% of tumor cells. Having said so, Engelman from Harvard have used a CR technology to use it in the clinical setting for non-small cell lung cancer, and both for EGFR mutated lung cancers as well as ALK mutated lung cancers. So if you are using it in the low passages, which obviously in the clinical setting it will be used, it may not be that much of a problem as long as you have a 90% of tumor cells.
What is the success rate for establishing tumor and normal CR cultures?
So it depends from tissue to tissue. In breast and lung, as an example, is almost 100% success rate. For salivary tumors that I have shown example today was the 100% success rate there too, but only 2 out of the 9 cultures were able to passage beyond 5. So in that, I would say the success rate is much lower, even though it was able to establish the cultures. So in general, the success rate is almost 80 to 90%.
Can low-grade tumor cultures be established from CR technology?
Oh, that's a very great question. Yes. Yes. So conventional, like I said earlier, the conventional cell lines are all from high-grade tumors. So it is really important to know whether we can grow the low-grade tumors or not, because that is where the need is. Yes, it does very nicely and very robustly. It creates the tumor cultures from low-grade tumors.
Is this technology applicable only for epithelial cells, or can it be used for non-epithelial tumors?
So yes, this technology was established for the epithelial tumors or epithelial tissues, but recently my lab has used this technology to establish cultures from melanoma and from brain tumors, which are non-epithelial. So it is unpublished work from my lab, but yes, it can be used. And actually for brain cancer, we have published one paper. Yes, it can be utilized. You have to tweak it. You have to change quite a bit in the media conditions, but it can be done.
And we have one last question that just came in. Has it been used clinically by other groups other than Georgetown?
Yes, it has been used very successfully by by Engelman group at Harvard, as I mentioned earlier, for non-small cell lung cancer patients. And I believe he has been moving forward to use it for other tumor types also at Harvard.
Okay, it looks like in the interest of time, we'll have to stop here today. Once again, I'd like to thank Dr. Agarwal for her presentation, and I want to thank all attendees for joining us today. If you would like to watch this webinar again, the recording will be available at gmosweek.com. Also check out gmosweek.com for more activities, including webinars, exclusive promotions, multiple chances to win a $5,000 credit for GeneWiz services, and much more.